Hey Team Bio, welcome to your screencast, Protein Synthesis and Secretion. In this screencast, we're going to kind of bring together two things that we are currently studying with something that we've already studied. Okay, we're going to be talking about protein synthesis and secretion. So you all know that proteins are like the action organic compounds of your body. They're responsible for, they make up enzymes, and they catalyze chemical reactions. The liver lab that we did, we were using catalase proteins in order to break down hydrogen peroxide, and that's when we saw the bubbles. Okay, so proteins play a super important role in living things, and they need to be made and then brought to the place where they're going to be used. The synthesis part we call kind of the central dogma of biology. It's how information flows from DNA to RNA to proteins. And proteins lead to traits and behaviors. Secretion is how those proteins get to the place where they need to get. So it's the movement of proteins through what we call the endomembrane system. And the endomembrane system is just all the true organelles that we've been talking about in class. They're the membrane-bound organelles. Okay, so this whole process starts out in the nucleus of the cell. The nucleus is full of DNA. DNA is what encodes your genetic information. It's made of 4 different nucleotides. And those nucleotides are A and T and C and G. A always binds to T, C always binds to G. That DNA is copied through a process called transcription. Transcription is when DNA is read and synthesized from that is an RNA. An RNA is also a form of nucleic acid, so both DNA and RNAs are nucleic acids. And this specific type of RNA is called an mRNA. It's a messenger RNA. It's going to bring the message of what's encoded in the DNA and bring it out of the nucleus. So the mRNA is exported out through a structure called the nuclear pore into the cytoplasm. So the message moves from the nucleus, the nucleus here, out of the nuclear pore, and now it's in the cytoplasm. It's still an mRNA. And now it's going to undergo this process called translation. Where the RNA is going to be read, and from that RNA, we're going to make a protein. In the cytoplasm are going to be a bunch of amino acids. They're going to be a little bigger than that. Let's see. So these are showing all my amino acids. And my ribosome is gonna run down my mRNA and read the message encoded in this messenger RNA. It's gonna grab amino acids from the cytoplasm and it's gonna string them together to form a polypeptide. In order for this polypeptide to become a protein, we have to fold it. And that's going to be polypeptide. So you all know that a protein has to be folded in a very specific shape in order to be functional. So in order to go from its linear structure to secondary, tertiary, quaternary structure, we've got to fold it up. That's going to happen in the rough endoplasmic reticulum. So here is kind of the view. So out here we have cytoplasm. And here's in our blue our ribosome. And here's our polypeptide. And you can't see it, but the mRNA message is going to be outside being read by the ribosome. And then, again, we've got our amino acids out here as well. Okay, so what happens here is that the ribosome attaches itself to the rough endoplasmic reticulum, or RER, and it shoots the polypeptide that it's synthesizing into the inside of the rough endoplasmic reticulum, where it's going to be folded. And then it's going to be a little piece of the rough endoplasmic reticulum is going to pinch off, and it's still carrying the folded protein inside of it. It's going to become a vesicle, a transport vesicle. Oops, see, it's LE, VES, transport, is LE, vesicle, okay. Okay, so you can see that this little kind of zoom in box was showing us this particular ribosome attached to the rough endoplasmic reticulum. And this transport vesicle is the same transport vesicle here. So the transport vesicle is going to take the proteins which are currently immature it's going to take it to the Golgi body. And this vesicle is going to fuse with the Golgi body and dump its contents inside the Golgi. So now the proteins that were in the rough ER are now in the Golgi. And the Golgi is going to sort the proteins. It's also going to modify them. So by the time transport vesicles bud off on the other side, we have mature protein leaving the Golgi. Now, these vesicles could be destined for a bunch of different places. This is also going to be mature proteins. Maybe this vesicle is headed to the mitochondria because it's full of proteins that are going to be used there. And this transport vesicle might be headed outside the cell. So this vesicle is carrying proteins that when it fuses with the cell membrane, it's going to release its proteins extracellularly. So this is the cell membrane. And as soon as this vesicle fuses with the cell membrane, this is now going to be extracellular proteins. This is just a fancy way of saying outside the cell. Okay, so this protein that is being secreted outside the cell, this could be catalase protein. Catalase is produced in your liver cells, but then it's exported to your bloodstream. So if you get a cut and then you put some hydrogen peroxide on that cut, it's going to fizz because catalase protein is the information is stored in the DNA. It's sent out as a message to the ribosome. The ribosome is going to synthesize a protein, which it's going to send to the rough endoplasmic reticulum. The rough endoplasmic reticulum then sends it to the Golgi body. The Golgi body is then going to sort it, modify it, mature it, and then export it outside the cell, where it can do its job of decomposing hydrogen peroxide into oxygen and water. Okay, that's all.